everyone, welcome to this week's tutorial for our kind of Excel exercises. This should hopefully be a super quick video as a lot of the things we're going to kind of run through today are things that we've done pretty recently in the past for some of um our weeks of our labs that we've done the last couple of weeks. And so it should all remain fairly familiar, at least in terms of like the graphing that we're going to be doing. So we won't dive into that a whole lot. The main purpose of this is just, again, to kind of run over how we're going to do these t-test comparisons. Um, Using the two different data sets that we have available, right? So that's going to be our flour data and our anthocyanin data. You'll see that the spreadsheet I send out to you guys has two separate sheets, one of which has our class flour data and the other of which has our anthocyanin data for both last week's lab, so week 5, and also our this week lab, week 6 as well. So that would be our pre and our post kind of treatment collection time points, basically. So, the first thing we're going to get kind of started doing is just by calculating these kind of summary statistics. We've done this several times already in the past before, and this is actually not something that's super relevant for the t-test comparisons that we're going to be doing, but we're just going to run over this super quickly, as like a quick reminder for how to do this and how to generate those plots, basically, right? So, again, we're just using the average function. We're starting out in this case with our flour data, right? And we're going to select the relevant kind of flower column. So we're looking at our purple control in this case. We're going to find our purple control. I'm going to select the relevant cells. We're going to close out those brackets, and there's our average calculated. We're then going to do this across our other kind of treatment and genotype combinations, as well as calculate those standard deviation values as well. And then once we have that, we should be ready to make our plot. Okay, so now that we have our mean and our standard deviation error values calculated, we're going to generate the same type of plot that we did last week with this flower data. That being the kind of paired bar chart, right? And so this is again really, really easy to do. Just go to insert up above, and then we're going to select this 2D column that's paired. And from here, we should be able to add those customized standard error bars the same way that we've done in previous labs, and then also to play around with our chart titles and our axes labels as well. Of course, if you guys are confused about how to do any of that, you have for your reference those past tutorials, which should have all that information. So hopefully you don't run into too much issue, but definitely reach out if you do. So let's now talk a little bit about how we're going to do these t-tests. These are actually really, really easy to do. There's no need to worry about implementing that t-test formula that we saw in some of our slides for the lab. It's all kind of built into an Excel function in this case, right? And so just some quick reminders about the types of comparison that we're doing. There's basically two pairs of t-tests that we're doing for each of our data sets, right? For both our flour data and our anthocyanin data. The first of these pairs of t-tests is going to be comparing across our genotypes. So when we compare across our genotypes, we um, keep the basically control or cold treatment, we keep the temperature treatment the same, and each of these t-tests is going to be comparing our purple for control versus the green for control, or the purple for cold against the green for cold. So to kind of illustrate what that means, basically, we're using this t.test function, right? And this takes four arguments. The first two of these are going to be our two data arrays that we're selecting. And then the last two are going to be specifics about the type of test that we're doing, right? So let's start out first by selecting our purple control data. Remember, we're comparing within the control kind of treatment currently. So each of our arguments is going to be separated by a comma. So when you're finished selecting that first kind of data set, make sure you enter in that comma before you select the second data set, like I'm doing currently. So we've selected that second data set. Again, we're going to finish off with a comma, and you can see that the comparison we're making is purple versus green control, right? The next thing we're going to input is the number of tails that we're including in this t-test. So for all of the t-tests we're doing, these are going to be two-tail t-tests, meaning that we're just comparing and testing for overall difference in the distributions rather than some specific directionality that's implied. So we specify that two-tail t-test by inputting a 2 here. And then that last argument is just going to be some assumptions we have about the variance within our two different sets of data, right? In this case, we're assuming that the two different sets of data have unequal variance, and they also don't overlap at all. They're completely separate. So we're going to input a 3 there for two sample unequal variance data. So once we've done that, we can close out those brackets and calculate what that t-test should be. And this is our first t-test value that we've done and completed now. We'll then repeat the same process for our cold data. What we're going to do now is, again, select that first array, this time looking at the flower data for our cold treatments. First our purple, followed by our green, followed by 2 for two-tailed t-test, and then 3 for that two-sample unequal variance assumption. So, and now we have our two t-tests that are comparing across the genotypes done. So remember, the key thing when interpreting these is you're looking for this calculated p-value to be less than 0.05, right? So anything that's less than anytime you see a p-value less than 0.05 indicates a comparison which is statistically significant, meaning that like the two sets of data that are going into this come from different distributions that are statistically different, essentially, right? So in this case, we would say that our purple control and our green control are statistically significantly different, basically, as compared to our cold data, which suggests that our purple cold and our green cold flowering data actually are not statistically significant and appear to belong to roughly the same distribution, right? Or could be drawn from the same distribution. Okay, we'll then repeat the same process for comparing data across treatments now. Now we're going to hold our plant genotype consistent, but basically select different columns of data depending on the specific treatment that we're interested in. So for our purple data, we're again start off by doing the t.test, and then we'll select our two data sets involved, right? First of all, our purple control, comma, then our purple treatment, comma, then 2 again for two-tailed, and then 3 again for unequal two-sample variance. We close out those brackets, and there is our p-value now for that third t-test, and we do the same thing for this last kind of comparison we're doing. So t.test, and selecting out that data, we're going to have our first kind of set here, our green control, comma, then our green treatment, comma, then 2 for two-tailed and 3 for two-sample unequal variance. And there we go. That is now our set of complete t-tests that we would be looking at. And remember, interpreting these is simple in the sense that we're just looking for any of these p-values that are less than 0.05, right? So in your grade scope questions, when you are talking about like differences in the reproductive fitness that we observe across these different genotypes and across these different temperature treatments, we'd be focusing specifically on where we see significantly different kind of comparisons, right? Where we see these p-values that are less than 0.05, essentially. If we look over at our anthocyanin data, we would basically repeat much of the same process that we did before. So again, we'd start out by calculating our means and our standard deviations, both for our pre and our post-treatment data. Whenever we work with our pre-treatment data, we're going to work with this column right here, our pre-column, and our post-treatment is of course going to come from that post column. We're not going to worry about generating any plots for our anthocyanin data. So once we've calculated those means and standard deviations, what we can move to then is just calculating those for t-test comparisons, right? And again, when we are comparing across genotypes, we hold the control consistent and we select the two different genotypes at that control treatment, right? So this is like for this first kind of t-test comparison we're doing, where we're comparing across the control, we would select for our first kind of data set the purple control values, and then for the second data set, the green control values, and then everything else just kind of remains the same, right? It's still 2 for two-tailed t-tests, and then that fourth argument is going to be 3 for unequal two-sample variance. We'd repeat the same process for our cold comparisons, right? Just shifting down, selecting that cold data. And then when we think about comparing across our treatments, we would select um, the same genotype data across the two different temperature treatments. And again, interpretation is the same in the sense that we're looking for these p-values that are less than or equal to 0.05. Anywhere we see a p-value that's less than or equal to 0.05, that's indicative of a comparison that is statistically significant, right? So when you're answering those grade school questions, focus on where you see those statistically significant comparisons. Okay, that's pretty much all we had to cover today. As usual, if you do have any questions, please feel free to shoot me a Canvas message, or I'm always available by email as well. I'm always more than happy to help. And yeah, I'll see you guys next week. Hopefully you guys found this a little bit helpful.